Hi there, I'm Bella, your Croyez AI reading assistant. Let's start today's reading journey. A trainer, IL-18 enhances the battle effectiveness of NK cells against cancer cells. How does IL-18 influence inflammation and immunity? Adipocytes and macrophages are among the cell types that constitutively produce interleukin-18, IL-18, a strong pro-inflammatory cytokine. The enzymatic cleavage of pro-IL-18, its precursor form, by caspase 1 within multi-unit complexes called inflammasomes is the process by which it is activated. For IL-18 to have its active effects, this activation stage is essential. A negative feedback system governs the levels of IL-18 after secretion. In response to increased IL-18 levels, more IL-18 binding protein is synthesized with the goal of binding and inactivating IL-18 to stop unchecked pro-inflammatory activity and possible tissue damage. The way that IL-18 affects biology is by attaching itself to a receptor complex that consists of two subunits, IL-18R-alpha and IL-18R-beta. The intracellular TIR signaling domains are present in both subunits. Remarkably, the receptor's beta-chain intracellular signaling can only be activated by the free component of IL-18. The increase of IL-18R-alpha occurs when naive T cells are stimulated with IL-12, making them more susceptible to IL-18 signaling. IL-18 has a variety of downstream effects that affect immune responses. It stimulates the expression of cell adhesion molecules, increases the maturation and activation of T cells and natural killer cells, and facilitates the synthesis of different cytokines and chemokines. Crucially, depending on the cytokine environment, IL-18's effect on CD4 plus T cells changes, inducing both Th1 and Th2 responses. It is noteworthy that this dual influence can modulate inflammatory processes. How does IL-18 influence NK cells and tumor immunity? NK cells play a crucial role in tumor immunity, recognizing and eliminating tumor cells, with their activation and expansion regulated by IL-18 through IL-18 receptors. IL-18 not only enhances NK cell expansion and cytotoxicity against tumors, but also induces an APC-like phenotype, expressing CD80, CD86, HLA-DR, and HLA-DQ. This unique NK cell phenotype acts as a bridge between innate and adaptive immunity, supporting the proliferation of tumor-specific killer T cells, reinforcing the significance of NK cells in tumor immunity, as highlighted by the success of PD-1 immune checkpoint therapy. Furthermore, IL-18 plays a pivotal role in accelerating the IL-2-induced expansion of human NK cells, offering potential improvements in immunotherapy. The synergistic effect of IL-18 with IL-2 presents a promising approach to enhance NK cell-based cancer treatments, addressing challenges in generating large numbers of therapeutic-grade NK cells. While IL-18 shares preclinical anti-tumor efficacy with IL-12, its clinical evaluation is limited. Nonetheless, the recognized potential of IL-18 in combination therapies, particularly in conjunction with IL-12 and IL-15 to stimulate NK cells, showcases its versatility. The cooperative effects of IL-12, IL-15, and IL-18 in generating memory-like NK cells hold promise not only for cancer immunotherapy but also for addressing various diseases, as seen in encouraging clinical trial results for relapsed/refractory acute myeloid leukemia.